Hi everyone, I'm excited to be presenting for the first time to the Animal Behavior Society, and I'd like to thank Esteban for organizing this symposium and giving me a chance to contribute. I'm going to use the next 20 minutes to talk about how we can leverage measurable properties of the body to better understand the sensory system and the visual system in particular. I'll begin with an overview of the current approach most of us use to study visual circuits in the brain, and then I'll tell you about some ideas and recent findings that have made me decide to go in a parallel direction that explicitly incorporates bodily movements. The modern study of the visual system, and here I mean the invasive study of visual circuits, is largely focused on understanding how visual inputs are translated into electrical activity by neurons. For inputs, we use highly controlled stimuli shown to the sensory receptor, which is the retina in the back of the eye. We do this by using a monitor placed in front of the eye. Then we measure the neuronal activity that appears at the same time using a variety of methods like calcium imaging. And then we look at the relationship between these two signals to try to understand what the neurons care about. And this is worthwhile because understanding this input-output transformation gives us some clues as to what the brain is doing. And we've learned a whole lot about the organization and function of the visual system this way. For instance, we know that neurons care about small regions of the visual field projected onto the retina. And we know that neurons that care about nearby regions of the retina are physically near each other in the cortex, so that the pattern of retinal activation matches nicely with the pattern of visual cortex activation. Another major discovery has to do with how the sensitivity and organization of neurons relate to the pattern of visual inputs experienced by the animal. We now have a lot of evidence for the efficient coding theory, the gist of which is that neurons don't waste energy computing irrelevant inputs. Another way of saying this is that a neuron sensitivity is going to be shaped by the inputs that the animal receives over its evolutionary and developmental history. A nice example of efficient coding has to do with color sensitivity in the mouse visual system, where the color preference of neurons here in the mouse primary visual cortex is related to their preference for the lower or upper region of the visual field. And the particular pattern with neurons that prefer lower regions corresponding to green colors and neurons preferring higher regions corresponding to ultraviolet wavelengths is consistent with both the sensitivity of photoreceptors in the retina and with the general properties of the mouse's natural environment. And this has actually been measured. Researchers use low-lying cameras to measure the wavelength distribution in the mouse's natural environment and confirm this hypothesis. So this type of coding is efficient in the sense that neurons that respond in the lower visual field spend more of their energy translating the frequent and behaviorally relevant green inputs than the rare and irrelevant ultraviolet inputs. And this is, of course, familiar to many of you. This is really a reformulation of similar ideas about biological structures being a function of an animal's unique ecological niche. There's a long and rich history of comparative analysis of retinal organization in different species and comparing it to their natural habitat. So here you're looking at the density of retinal ganglion cells in different species and a sample of what their natural visual input would look like from their perspective. As an example, it's been hypothesized that this increased ganglion cell density in the lower retina of mice, which projects to the upper visual field, helps mice detect and avoid overhead predators. And the takeaway here, again, is that even at the very first stage of the visual system, the retina, the structure and function seem to be optimized to the pattern of inputs that are received. So this has been successful, and we've learned a lot about visual and more generally cortical computations using the modern systems neuroscience approach. But of course, this method has some trade-offs. In order to deliver precise visual inputs and measure outputs, we tend to immobilize the animal. This limits our access to behaviorally relevant context And it has a confound that animals will attempt to move even though we ask them nicely not to. So our sensory responses are contaminated with other signals. And what this produces is basically a disembodied brain without species-specific flavoring. So we can swap out the mouse for a cat or for a human and largely run the same experiment. And I think we all agree that this isn't ideal, but some of you may be thinking that incorporating the body isn't super critical if all we care about is really just the sensory input-output transformation. So I want to take a minute to go into the specifics of why, even for studying just that transformation, the body plays a major and potentially unavoidable role. First, bodily movements result in major changes to sensory input. We call this self-generated sensory feedback, reafference for short. So when we move our eyes, heads, and bodies, the part of the visual field that's projected to the retinal image, can change drastically. And we usually don't perceive reafference, but it's not just an artifact that the brain learns to remove. It's useful to the animal and it's behaviorally relevant. For instance, optic flow is the visual reafference produced by locomotion. So as I move forward in the world, the visual flow on my eyes is called optic flow. And we use it for balance and for posture correction, for driving, and it acts as a sort of visual proprioception that helps us locate our bodies in space. And going back to the fish encoding idea, neurons are likely shaped by reafferent sensory inputs because they're very frequent, they're behaviorally relevant, and they're important for survival. So if we want to understand neural processing, we need to measure and understand self-generated sensory inputs and not just focus on external environmental inputs. But movements don't just influence the distribution of sensory inputs. We have some evidence that movements actually change sensory processing by setting off a closed-loop signaling cascade. So when we isolate the retina and the brain and present external inputs to an immobilized animal, we're studying a small piece of a larger integrated system that may not be separable. So let's go through this system. Self-generated movements are a result of a motor command from the motor system to the spinal cord that moves the body, or more specifically, the sensor that's on the body, from an initial position to a new position. And this change in sensor position and say eye position produces reafferent sensory input that we've already talked about. At the same time, though, a copy of the motor command is used to predict the new position of the sensor using an internal model. And then this prediction is used to predict the resulting change in sensory input. So self-generated movements produce both reafference and a prediction of the reafference. And the resulting neuronal output then is produced by subtracting the predicted reafference from the actual reafference. And this discrepancy is a type of prediction error that we think is fed back to improve the position predictor and the sensory predictor models. And we have some evidence for different pieces of this signaling cascade, and in the visual system, a lot of it comes from studies in the flies. But it's very complicated, likely distributed, and still largely an open question. And there's also this interesting challenge in going from the predicted sensor position, which is in the reference frame of the muscles that cause body or eye movement, so in motor coordinates, to the predicted sensory input, which is in the reference frame of the retina, and we don't know exactly how that's done. But all of this paints a pretty compelling picture for a major role of reafference in the sensory motor system. Being able to predict the sensory consequences of our movements is critical not only for survival, but also for our sense of self and agency. And because we're moving constantly, the brain needs to make constant predictions of reafference. So considering efficiency, a very reasonable hypothesis, I think, would be that brains are optimized to predict reafference. So in other words, every element of the signaling cascade, both the motor and sensory system and the models, have design elements that make the prediction of reafferents more accurate and more efficient. And now we can't directly measure the internal models because we don't understand their underlying circuitry, but we can access the motor and sensory systems and test the unifying hypothesis that their structure and function is optimized for the prediction of reafferents. So how do we test this reafference hypothesis? Well, it's not easy, but that's what long research programs are maybe for. So we have to start with behavior and measure the self-generated bodily movements. We also have to measure the resulting reafferents. And once we have these signals, we use a computational model to predict one from the other. And once we have this trained model, then we can use it as a testbed to produce hypotheses for which aspects of sensory design will improve the prediction of reafferents. And finally, we can check these intuitions gained from the model by actually measuring neuronal activity in freely moving animals as a last step. So some of you might be thinking that all this sounds nice, but it requires leaps in technology we don't have yet. So I'm going to spend the next few slides telling you that we do. Let's go through step by step. We can measure eye, head, and bodily movements using wearable sensors and cage-mounted camera recording systems. So here you're looking at a very light camera-based eye tracker system that's worn on top of the rat's head. It's basically 2 miniature cameras that use infrared light reflected through these 2 little mirrors to record the rat's eye. The mirrors pass visible light though, so the animal still sees the full visual field. And here's what that system looks like in a freely moving animal that's hunting a cricket. You can see the videos are stable, and you can extract the position and size of the pupil with a few lines of code. For movements of the head and other parts of the body, inertia sensing chips and camera-based systems can both capture the position of various body joints with fairly high resolution. So that's how we record movements. How do we measure the resulting sensory reafferents, the visual inputs generated by these movements? This is a bit harder, but a couple groups have already solved this problem using either a head-mounted camera that couples with the eye tracker or a model of the environment that couples with the orientation of the body and the eye tracker. So here's an example. This is a model of the environment that the rat is in. I think this is a mouse actually, and that's coupled with the eye tracker to give you the actual retinal image or an estimate of it that the mouse sees as it's hunting a cricket. So you're looking at both internally generated sensory inputs and externally generated inputs. And notice how in the end here, as we get close to the cricket, it lands sort of on the stable part of the retina. See the retina stabilize for a second. So that gives us both movements and a pretty good estimate of reafferent visual inputs caused by movements. The next step is to then take this pair of movements as inputs and reafferents as outputs and use them to build a model that can predict one from the other. And deep learning models are perfect for this sort of paired problem, specifically when there's a lot of data. It's not trivial because we would need to make sure the models are biologically plausible, and that they translate visual inputs in a similar way as real brains. But this has partially also been solved because deep learning and other types of these models have already been used and implemented in autonomous robots who face a similar problem as animals, that they have to predict the sensory consequences of their movements. And one way the robots learn this is through a process called motor babbling, where the robots initially move their joints randomly and record the sensory inputs, and by iterating this process, they improve their internal model. And once we have a fairly accurate model, then the real work begins. We can use it as a test bed to determine which aspects of sensory circuitry are critical for cheap, fast, and accurate predictions of reafferents. So now we build real hypotheses, and finally, we use wireless loggers that are available off the shelf these days and very light to record neural activity to test the hypothesis we formed with the model. in a way that doesn't impede the movements of the animal. So let's talk about a specific example. We'll go back to the species-specific retinal organization case again. Most previous attempts, not all, but most, to explain the retinal organization have invoked sensory inputs produced by the natural habitat. But these are hard hypotheses to test because it's hard to measure sensory inputs produced by an animal's natural habitat if you're in the lab. But it's easy to measure sensory inputs produced by bodily movements. So can we use bodily movements in addition to or instead of the natural habitat to predict the retinal region with the highest density of cells under the reafferent hypothesis? So let's just try to reason through how retinal organization could improve the prediction of reafferents. Well, the densest region of the retina is a first approximation, also the most computationally expensive piece of the tissue. So if we wanted to optimize for cheap prediction of reafferents, it would make sense to have this dense region in the part of the retina that experiences the least amount of reafferents, that requires the least prediction. In other words, the most stable part of the retina during movements should be where the highest density region falls. And we don't have much data on this, but a recent paper from Holmgren et al. Basically showed this in the mouse retina. This is that same paper that used that actually estimated reafferents during cricket prey capture. But there's a twist. So the most stable part of the retina here during prey capture is this region with the red cricket. And you can see that it doesn't overlap with the high density ganglion region in the top. But it does overlap with a particular subtype of retinal ganglion cells that are sensitive to small, fast-moving objects like crickets. So it seems that the organization of this particular cell type may be optimized to improve prediction of reafferents during prey capture. And we can now further explore this idea by looking at the connections of these cells to the rest of the sensory and motor system. But in order to test this hypothesis well, we ideally need measurements of movements and retinal organization from at least 2 animals who either move differently or have different retinal layouts. And this brings me to my favorite model organism, the tree shrew, which is particularly suited for exploring the role of movements in sensory system design. And that's just because they're highly visual animals. Their visual system is similar to primate in many ways, but they move in really elegant, acrobatic ways. And unlike rodents who move mostly, who crawl in 2D, and bats and birds who fully move in 3D, they're sort of in the middle. They can move in 3D and do long jumps, but they can also crawl. So for the past few years, I've been studying the visual system of these animals without regard to their bodily movements. And I've made some cool discoveries, but they've also really forced me in a way to consider how the tree shrew's behavior, and specifically movement, might influence sensory organization in all parts of the visual system. As I alluded to at the very top of the presentation, we know that neurons in the primary visual cortex in the back of the head preserve the layout of the retina, so that nearby neurons on the cortex represent nearby regions of the retina. This is the case in V1 and other primary visual areas of basically every animal studied so far. There's a simple mapping. But when I looked at the secondary visual cortex, this visual area directly adjacent to V1 in tree shrews, primates, and other animals, I found a totally different organization. In V2, neurons don't preserve the layout of the retina. And instead of a simple point-to-point transform between the retina and cortex, there's this complicated, snaky transform that results in striped maps of the horizontal dimension of the visual field. So if I wanted to activate V1 sequentially from side to side, the stimulus I would need to show the retina would just be a bar moving from left to right, and that would produce the same activation pattern in V1. In V2, that stimulus wouldn't be a bar, but it would be a dot that's moving like this in the visual field. So why would V2 have this very strange organization? One possibility that I'm excited to test is that it's somehow related to the scanning motion that tree shrews make with their heads. And I have some examples of this. These are fast, jerky movements that the animals use to explore their surroundings. Here's another one, you can see it well. So they're fast and jerky, and they can be up and down too, but they're usually more in this horizontal dimension. Now, this head motion might have nothing to do with the organization of V2, but I'd love to rule it out on my way to finding other explanations. And also getting the data to be able to test this hypothesis would be incredibly informative for many other questions about the role of bodily movements in sensory design under the reafference hypothesis. So to recap, the guiding question of my research program is to understand how the development and function of the sensory system reflects the body and bodily movements of the animal. And I think answering this question will really allow us to leverage the diversity of engineering and design solutions that nature has evolved to understand our brain as really a subset of a larger system that includes the body and bodily behavior, and ultimately also the environment. And just a couple advertisements. I'll be starting my lab next summer at the Cornell Department of Neurobiology and Behavior, where many of the faculty and students share deep interest in animal behavior. I'm hiring at all levels, so please get in touch if you're interested in this line of work. I also want to plug an upcoming meeting that I'm co-organizing focused on recent discoveries in tree shrew neuroscience and behavior that'll be held in Florida at my institute in September. You can join us. There will be a virtual option. You can join us if you're interested in tree shrews, and I really hope that some of you do, because there is a real gap of knowledge about tree shrew ecology, evolution, and especially behavior that needs to be filled. All right. Thanks for tuning in, and I guess I'll see everyone in a